Hello, it's 11 o'clock our time. I welcome everybody who has joined by now, and I think we can start. Welcome to Solis Biodyne webinar on Endpoint PCR. My name is Olga Kukk, and I am the head of R&D at Solis Biodyne. Today, here with me is our wonderful product manager, Ekaterina Nebogatova. With over 15 years of wet lab experience, she will happily share her thoughts today on how to make the most out of your assay. We will start with a 20-minute interactive talk by Ekaterina, followed by 10-minute Q&A session. And if questions will pop up during the talk, feel free to post them into the Q&A box, and we will go over them right after the talk. And before we dive into today's topic, I will take a few moments to introduce who we are. Solis Biodyne is a nearly 30-year-old biotech company located in Estonia. We produce high-quality PCR reagents and we provide them to customers all over the globe with both business-to-business solutions and, of course, end-user directed solutions as well. Solis Biodyne products are well known for their excellent stability. This we achieve by incorporating our unique patented stability tag technology into every protein we produce. And now, without further ado, I'm happy to give over to Ekaterina. Take it away.
And thank you, Olga, for this nice introduction. And without waiting any longer, let's dive into the topic of today's webinar and find out how you can master endpoint PCR by optimization and creative problem solving. PCR polymerase chain reaction is invented already about 40 years ago, and it's still a gold standard. Depending on the aim of the assay, other modern techniques like isothermal amplification, NGS, CRISPR, etc. can be applied. Each type of technique has its own benefits, but it also has its limitations. Most new generation techniques still require significant investment into the equipment and time for personnel to learn and master them. On the other hand, classical PCR is a common technique, and it's easy to find compatible equipment in most molecular biology labs. Different software are available for primer design. PCR reagents are produced by a lot of vendors and also different software available for primer design. All of that makes implementation of PCR into your routine lab work reliable strategy. Endpoint PCR is still the most straightforward choice for such techniques as cloning, genetic mutation, forensic analysis, pathogen detection, and genetic diversity studies. But let's find out what are checkpoints. So endpoint PCR assay planning checkpoints. Starting new project can be a challenge. Let's go through 3 points which I have compiled. First, make sure you have access to all equipment that is required for the assay. You will need at least thermal cycler, micropipettes, consumables like tubes, pipette tips, etc. And you have to think about what kinds of post-PCR analysis you want to perform. So depending on that, you will need either equipment for agarose gel electrophoresis, or maybe you want to do sequencing or mass spectrometry. You have to think ahead, what will you need? The next checkpoint is assay design. When the target is chosen, it's time to design a couple of pairs of primers. We strongly recommend trying out at least 2 or 3 pairs of primers. Depending on specific assay requirements, do not forget to include controls already on stage of planning. Design can be complicated if you choose to perform multiplex reactions, but modern software can help you out with the design. At this point, you already know that post-PCR analysis methods you will perform. You have chosen it. So do not forget to write down requirements for that as well. Now you have identified your precise needs, and it's time to find right reagents to meet all of them. It's a final checkpoint in the planning process for endpoint PCR assay. And as many scientists tend to stick only for one familiar brand to work with, even though we usually state that we want to explore everything new. When it comes to our reliability, we choose familiar ones. So challenges with less familiar products can be discouraging. And it's time for our first poll. Please share with us what is the most challenging for you. Is it finding a reagent that meets your requirements? Or maybe it's optimization? Or maybe it's waiting your ordered reagents to arrive, or maybe you are hypnotizing bands on the Agile scale. If you stare at them, maybe they will run faster. Let's find out what everybody is thinking. Looking like optimization is most challenging for you, and finding the right solution is not so much. So, seems everyone has his own struggle. To help you overcome some challenges, I would like to share 3 tips that can help you with optimization of the assay and workflow in general. And first tip is using pre-mixed master mixes. PCR reaction requires at least DNA polymerase, buffer, magnesium, DNTP, water, primers, templates in reaction mixture. It's a lot of pipetting. And so almost all the liquids are colorless. You can forget to add something or add something twice. Also, pipetting error is accumulating, so reducing pipetting steps significantly reduces error rate. Another benefit of master mix is that in storage and maintenance of fewer amount of vials is much more easier. Compare like 15 or 5. Makes sense. Next tip is relevant for those who are performing post-PCR analysis via visualization on agar gel. Back in the days, it was year 2008. My first experience with Solis Biodyne was making a colony PCR. I made it with Fireball master mix, ready to load. Here in the right bottom, you can find mixtures that we have ready to load solutions. And I had one with Fireball master mix ready to load. Just picked a colony, stick into the mixture, and this went straight to the thermal cycler. After PCR, I did not any additional purification step and made a restriction analysis with results of PCR reaction. And this mixture went straight to the agar gel. It was easy, fast, didn't require a lot of pipetting steps, and it worked really well. And with ready-to-load master mixes, everything is already in the solution, including loading dies. So you can easily track moving of our bands on the gel. Those mixes does not inhibit reaction. Actually, we even received the feedback from our clients that ready-to-load compositions from master mixes are working better comparing where the loading die is not included. We have some speculations about what is causing this phenomenon, but it will be interesting to find out what you are thinking about that. So we encourage you, maybe in chat session, you can, in the chat box, you can ask or share your thoughts. Moving next. Third tip is perform multiplex reactions. That's where you can get more data from the same volume of the sample, which is especially needed when you work with not so much of starting material and you don't want to lose it. It's saving time, labor, resources. In my labs, we had to book for PCR thermocycle while I had to get the chance to run our assays. It's much more easier to find time for running the one reaction instead of 18. If you are working with several templates, crossover contamination risk is a real threat. And reducing amount of tubes and pipetting steps is a good addition to your contamination prevention strategies. Planning ahead is the best way to get reliable results fast. So now we have discussed 3 tips. Implementation of master mixes, using ready-to-load solutions, and multiplexing. Here in right bottom, you also can find some our mixes which can be used for applying all those 3 tips. So we have discussed those 3 options for optimizing, but what do you think? Are there something else? Here is time for our second poll. Please choose. What's the option are the best practice to reduce expenses even more? What do you think? Is it using high quality reagents or so we can get reliable results from the first attempt already? Or order reagents that are shipped at room temperature? Funny it is, but often cost of the shipment on blue ice is much higher than reagent itself. Or best strategy is reducing the runtime. It's reducing labor expenses, and labor expenses are usually the biggest part of the budget of the project. Quite hot topic. Everyone wants to get the results fast, easy, reliable, and as cheap as possible. So the most popular was high quality reagents, manage shipping at room temperature, and reducing reaction time. We can all agree that every piece of advice is valuable when it comes to reducing costs without sacrificing reliability of the results. We want our results to be trusted. Let's find out how Solis Biodyne ensures that our products perform the best possible way. Stability tag. It's what pops up when we are talking about Solis Biodyne. Stability tag technology is our signature, which is implemented into each protein produced in-house. On the right, you can see the structure of Fireball DNA polymerase with integrated stability tag. Stability tag is in yellow. Thanks to that technology, we are able to ship all our products from the catalog at room temperature in the envelope. That's significantly reducing shipping costs. Stability is guaranteed for at least 30 days, even when exposed to room temperature. And that's not all. Even more, stability tag ensures remarkable long-term stability. Here at the bottom left, you can find an example of Fireball DNA polymerase stability. Even after 20 years, it remains 50% active. We still have small aliquots from this first batch and somewhere in our fridges. And we plan to add additional data point next year. Besides high stability, what benefits Solis Biodyne solutions can offer? Please welcome Solis Fast product range. With products from this range, we can complete a PCR reaction in just 15 minutes. Those results were achieved on Eppendorf-Meister cycle X50 test for amplifying 50-100 base pair DNA target. Drawing on our experience, we have incorporated all these positive features into the master mixes, which also offer multiplexing capability and ready-to-load options. However, it's not just about speed with SolidFast. It's also perfectly complements your carryover contamination prevention strategy. Our portfolio includes mixes with 100% DE2Ps and UNG, ensuring peace of mind and eliminating concerns about the threat of carryover contamination. This does not impact the reaction or Sanger sequencing. Dealing with contamination can lead to halting all activities, lab cleaning, days, weeks, and you have to retest all samples that were analyzed between contamination checks. Something everyone wants to avoid. Sometimes it's hard to believe that all of that is true. So let's discover how scientists from around the world are benefiting from our reagents. Let me share. Here is our website. If you go here, choose products, Endpoint PCR. And here we can find the whole portfolio for our Endpoint PCR mixes. Now let's choose my perfectly favorite. For example, hot fireball plant master mix. It's our rockstar. It's most popular among our clients. Here we go to the product page. If you go scroll down, you can find here 220 citations. Choose some of them, a lot of fresh ones, and just click to read article. You can read it on our website, or you can go to the BIOS website. All articles are totally available for reading for free. So you can find a lot of different applications there, protocols, whatever you are interested in. We have come to the end of this webinar segment where I'm sharing with you. And in the summary, let's outline key take-home messages. First, when initiating a new project, the first steps involves planning. Document all your objectives, template selection, and the necessary equipment and reagents. Next, while assay optimization, consider reduction of the steps and reaction time by employing master mixes and protocols with fast enzymes and cyclers. Third, make sure you choose thoughtfully. Reagent must align with your specific requirements and come at a reasonable price, offering the best quality price ratio. So plan, reduce steps, and think ahead. Choose wisely. Not to forget, our hottest campaign is still running. To find out more information, you can visit our website, solidbiodine.com, or contact us in the info@solidbiodine.com. That is all from me, and thank you very much for attending and attention. And now I hand over it for Olga.
Thank you, Ekaterina, for a wonderful talk. And now I will go over the Q&A session. So for now, I see 3 questions. In case you feel others can still post them, but let's go over these 3. I'll just read them. Iris is asking, how well do your master mixes perform with complicated sample matrices? Can you comment, Ekaterina?
Yeah, I'm going to unmute myself. When you're talking about matrices, I guess we can translate it to the inhibitor tolerance. We have different feedbacks from our clients, especially about my favorite blend master mix, that it's really performing well with plant samples and soil samples, even though you can do purification steps, even though there are a lot of inhibitors remaining in the samples, and it's performing really well. We have performed in-house tests with SolisFast product range, but it was made for qPCR, but I guess we can translate it for endpoint as well. And they are performing really well with quite a large panel of different inhibitors like urine, PBS, and different transportation medias for swabs. I hope it's answered your question.
Yes, I think it should. So we have a few more questions in Q&A from Robin. Hi, Ekaterina. Thank you for sharing your thoughts on PCR optimization. Does the nucleotide dye already present in the Solis Biodyne mixes interfere with sequencing efficiency in the PCR fragments if the PCR fragments are sequenced using Sanger or Illumina or Nanopore, etc.?
Thank you, Robin, for your question. Yes, if you use ready-to-load mixes, it can affect sequencing results, but we have mixes without dye. So for every application, again, you choose wisely, think through what is your post-PCR analysis, and if you do not require to visualization on other scale, just use mixes without dye.
Yeah, exactly. And there's a plenty to choose from in our portfolio. Okay, next one up is Mikko. You mentioned carryover contamination. How common is this? And how well do your mixes actually protect against it?
Thank you, Mikko, for question. Lately, due to the COVID time, and we had to move fast, to learn fast, to find more employees which are not so much trained, let's say, and doing a lot of assays. That means a lot of amplification of the same target in the same lab, and maybe not so much cleaning every time, and maybe not so following the rules because you want a result really fast. So carryover contamination become really a struggle for a lot of labs. And our mixes are protecting it real well. SolisFast can implement 100% of DUTPs into the amplicon. It's totally compatible with UNG. And our UNG is really fast. It's, if you set up reaction at room temperature, it doesn't have to be an additional treatment step. So it protects it very well.
Thank you. I think that answered the question. Then Gani is asking, is saying, shipping in the envelope, ... How stable your products are at temperature higher than usual room temperature?
Yeah, thank you, Gani, for question. It is tested. Our usual panel includes testing at 37 degrees. Almost all our products are stable for 2 weeks at 37 degrees. Some of them even go further, and it has about 1 month of stability at 37 degrees. So it's totally dependent on the product. But we are testing at the higher temperatures as well to be able to ship to every country in the world.
Exactly. Thank you. And then I see we have a follow-up question, I guess, regarding to your comment to the carryover contamination. Anonymous attendee is asking, can you comment on the difference between a PCR primer and just a sequencing primer?
Yeah, when you are doing sequencing, let's say reliable results you can get about for up to 1 kb. But you can amplify the longer amplicon with PCR. For our mixes, it's about 5 kb. So when you're planning sequencing, I strongly recommend you to take that into account, that you cannot sequence for Sanger sequencing, you cannot sequence the whole 5 kb in one run. So you have to plan primers to the different spots of the amplicon.
Okay, for now, yeah, I think that should be. Oh, we have just the last. Let's take the last one from Q&A. Just popped up. Xander is asking, hi, Solid Green versus Eva Green. Can you say something about the sensitivity and specificity of these products compared to each other? And which would you recommend? That's always a good question.
Yeah, it's a really good question, but it comes down again for your application. And I think it would be wiser to answer you after this email by email, because it's there are some coming some graphs maybe and some comparison which are kind of hard to remember every detail for me right now. But yeah, they're slightly different, and depending on the application, there's different choices.
Thank you. And with this, we have come to an end of our half an hour. I thank everybody for attending. If you have any remaining questions, still feel free to reach out to us under info@solisbiodan.com or visit our website. You saw it in the talk. It's very easily navigatable. And I hope you enjoyed what we had to share with you, and hope to see you all in our next webinars. And then don't forget that the campaign is still running and you can still have a discount on Hot Fireball product. Thank you.
You.
And bye.
From you as well. Bye. Thank you.